You can use the chat, for example. Ну, я его включил вообще даже пожалуй, не может.
Да, сэр.
А. Так, ну да.
Он тут позвонит?
Да.
Я подбегу.
Good day, dear colleagues. My name is Elena Onischuk, and today I'm your lecturer, and our lecture is biological effects of radiation. If you're ready to start, we can start our lesson. Please, if you don't see the presentation, write it in the chat, and we try to improve our internet connection in our presentation. Okay, let's start. Content of our lecture today, you can see on the slide. We have a big lecture. Because this is a very, very big theme and very important. And I want to tell you the most important point of this theme. So we have about 2 hours or maybe more. And I hope you are ready. Okay. Radiobiological effects. What is that? Radiobiological effects, functional and morphological changes that develop in the body as a result of exposure to radiation. Biological effects of ionizing radiation different and depend on the type and the intensity of exposure. Biological effects of various radiations studied by radiobiology. According to the criterion of formation mechanisms, radiation effects divided into two types: target and non-target ones. Target effects consist of two groups: deterministic and stochastic. According to the new terminology of the ICRP, instead of term deterministic effects, the name tissue reactions is used. Radiobiological effects and consequences of irradiation divided into somatic, manifested in the exposed person himself, and manifested in his descendants. Somatic effects, in turn, divided into deterministic and probabilistic. stochastic effects. Deterministic effects are clinically detectable harmful biological effects caused by ionizing radiation, for which a threshold is expected to exist, below which there is no effect, and above which the severity of the effect depends on the dose received. Such significant effects manifest themselves in the form of obvious pathology, acute or chronic radiation sickness, radiation burns, so-called local radiation injuries, islands, cataracts, temporary or permanent sterility, and etc. These effects most often occur with short-term exposure to high doses and high dose rates of radiation and have a threshold character. For the occurrence of particular pathology, it is necessary to achieve threshold levels of radiation doses, below which these effects are not clinically manifested. Stochastic effects, distinguished by absence of dose threshold. It means that implementation of stochastic effects theoretically possible at arbitrarily small radiation dose and low dose less likely than occur. In the case of stochastic, probabilistic consequences of exposure, we are not talking about severity of lesions, but about an increase in the frequency of manifestations of cancer or hereditary defects in a group of people, including each average individual. Consequently, the greater the radiation exposure to a group of exposed people, the higher, other things being equal, the probability of exit frequency of long-term effects in individuals. For the deterministic effects, the dose-effect relationship has sigmoid form. According to a linear non-threshold concept, severity of effect manifests itself linear depending on dose. Higher dose, greater effect. And for the manifestation of these effects, there must be a threshold the International Commission on Radiology Protection, ICRP, adopt the concept in 1959. Stochastic effects usually detected long after exposure. Probability of their occurrence considered non-threshold dose function. The non-threshold concept was adopted as a working hypothesis by international scientific organizations to substantiate principles and methods for regulating low doses of radiation. So, the direct effect of ionizing radiation is such changes that occur as a result of the absorption of the radiation energy by molecules, and damaging effect is associated with act of excitation and ionizing of atoms and macromolecules. Indirect action of ionizing radiation changes in the molecules of cells and tissues caused by the products of radiation decomposition of the water surrounding these molecules and low molecular weight compounds dissolved in it, and not by the radiation energy absorbed by the studied molecules themselves. Based on the concept of the direct action of ionizing radiation, theory of targets and hits arose. This theory explained the presence in the cell of a vital center, a gene or ensemble of genes, a target hitting which one or more high-energy particles of atomic radiation is sufficient to destroy and kill the cell. Hitting a target is a probabilistic event. A further development of the theory of the direct action of radiation was the stochastic theory. It, like the target theory, takes into account the probabilistic nature of the radiation entering the sensitive volume of the cell. But unlike it, it also takes into account the state of the cell as biological object, a labile dynamic system. Under the influence of ionizing radiation, physicochemical processes beginning in the cell, as a result of which highly active chemical compounds, radicals, and ions formed the damaged biological structure of the body and cause a mismatch of its functions. Impact of ionizing radiation on biological objects generates a sequence of processes that are strikingly different from each other on a time scale. Oxygen effect manifests itself in all biological objects, microorganisms, plants, animals, and at all levels of their organizations, subcellular, cellular, tissue, organ, and organism, significantly weakening all radiobiological reactions, biochemical disorders, mutations, inhibitions of growth and development, and increasing survival of irradiated organisms. The mechanism of the protective action of hypoxia is explained by the fact that during irradiation, in the presence of oxygen, peroxide radicals are formed which enhance the effect of radiation on vital macromolecules and cell structures and or weaken the effectiveness on intracellular protective substances. The magnitude of the oxygen effect depends mainly on the type of radiation and conditions of the exposure. Greatest effect is observed under the action of X-rays and gamma rays. With an increase in the ionizing density, the oxygen effect decreases, and under the action of the most densely ionizing radiation, for example, alpha rays, is practically absent. In normally water-reactively vital biological objects, weakening of radiation occurs only when hypoxia is applied during irradiation. In dry objects, resting plant seeds, bacterial spores, and during hypoxia after irradiation, during the transition of irradiated objects to active life, for example, during seed germination, an oxygen effect used in radiation therapy, increasing the oxygen content in the tumor and creating hypoxic conditions in surrounding tissue. It is possible to increase the radiation damage in tumor cells, while simultaneously reducing damage to healthy tissues. Concept of occurrence of potential hidden damage was formed in radiobiology by the end of 1950s, influenced by the works of Alexander, Charles B., Gordy, Alper, Eidos, and etc. According to these views, latent damage occurs in macromolecules during their ionization, which in absence of oxygen do not in themselves lead to a loss of activity, but being fixed by oxygen turn into obvious damage. As can be seen, oxygen effective radiomodifier, it has radiosensitizing effect. Quantitative expression of the increase in radiosensitivity in the presence of oxygen is the oxygen enhancement effect. The value of this coefficient shows how many times there is increase in the radiosensitivity, decrease in radioresistance of living object in oxygen environment compared to their irradiation in oxygen-free conditions. For example, when irradiated a suspension of bacteria under anaerobic conditions, the value of lethal doses 50 equal 1000 gray. When this type of bacteria was irradiated in the environment with oxygen content, the lethal dose 50 for them was only 500 gray. The oxygen enhancement effect in this case will be equal to. It has been experimentally shown that participation of oxygen in the realization of emerging potential damage occurs during radiation. This introduction of oxygen into suspension of bacteria cultivated under anoxic conditions 20 milliseconds before irradiation enhances their damage. The addition of oxygen 10 milliseconds after irradiation does not change the damaging effect of ionizing radiation. The sensitizing effect of oxygen during cell irradiation is manifested only when it is present in the tissues at the time of irradiation. At present, there are no clear ideas about mechanism of sensitizing effect of oxygen during irradiation. The most generally accepted point of view is that oxygen molecules having electron withdrawal properties actively interact with resulting free radicals, including the radicals of biological molecules. As a result, new active free radicals appear and the potential damage to molecules resulting from irradiation is fixed. Damage fixation consists in stabilizing the damaged structure of molecule, which makes them inaccessible or difficult to access for the repair system. Lifetime of such oxygen-dependent lesions can be long. For example, in macromolecules, such damage persists for several hours. If the solution contains impurity molecules capable of competing with macromolecules for active product of water radiolysis, for example, or antioxidant, then this leads to decrease in radiation damage to macromolecules. In living system, taking into account only the oxygen content is insufficient. In this case, oxygen effect determined, for example, by the inactivation of DNA or phages in solution by cell death manifests itself only in the presence of SH compounds. The latter compete with oxygen for potential damage that occurs in DNA, determining their fate. Upon fixation with oxygen, they are realized in obvious damage. This phenomenon was first observed by Alper in 1952-1954. who discovered the protective role of oxygen during the inactivation of bacteriophage SH. So, temperature effect. Radiosensitivity of many macromolecules depends on the temperature during irradiation. Mechanism of the temperature effect not been finally established. It is assumed that the rate constant of reaction that determine the inactivation of macromolecules depend on the temperature. At least some of these reactions may be interactions of macromolecules with atomic hydrogen and other small radicals that released when organic materials are irradiated and likely attack undamaged molecules. Temperature after effect. Hidden damage occur in irradiated protein molecules, turning into obvious ones with additional thermal exposure, which is expressed in enzyme inactivation after heat treatment of the irradiated preparation. The thermal effect was also effective after aerobic irradiation, which indicates that another damage are realized by heating, different from those can be affected by oxygen. The presence of impurity molecules. Irradiation of protein macromolecules in a mixture with a number of low molecular weight substances can either reduce their radiosensitivity, protection effect, or increase it, sensitization effect. Substances containing a sulfhydryl group or disulfide bond have the most pronounced protective effect in dry systems. At the same time, the addition of sucrose increases the degree of enzyme inactivation, for example, ribonuclease by 2.6 times. A number of carbohydrates have a similar effect. Elucidation of the mechanism of protective and sensitizing effect was the subject of radiation biochemical analysis. A number of hypotheses about the mechanism of protection of macromolecules by low molecular weight compounds are discussed. First, their action may be due to competition for highly reactive free radicals, which can cause structural damage to the macromolecules. Another possibility is that the protective agent helps to repair the damaged macromolecule by transferring hydrogen from protective substance to the free radical of the damaged pH biomacromolecule. Sensitivity of different biological materials to the action of ionizing radiation significantly different. Radiosensitivity and radioresistance are concepts that characterize the degree of sensitivity of animal and plant organisms as well as their cells and tissues to the effect of ionizing radiation. The more changes occur in the tissue under the influence of radiation, the more radiosensitive the tissue, and conversely, the ability of organism or individual tissues not to give pathological changes under the action of ionizing radiation characterizes the degree of their radioresistance. In oncology, radiosensitivity and radioresistance refers to the rate and degree of tumor response during treatment. Radiocurability reflects the extent to which the assumption of tumor eradication is realized and subject to limitations such as resistance of normal tissue. These two properties do not necessarily correlate. Tumor may be highly susceptible but radio incurable. For example, leukemia and myeloma, or relatively radio-resistant and radio-curable. An example of tumor that simultaneously exhibits the properties of radiosensitivity and radiocurability is lymphogranulomatosis. Renal cell carcinomas and malignant melanomas, radio-resistant and radio-curable. Different organisms, as well as different organs and tissues of all plant and animal organisms, also have different radiosensitivities. Sex cells and white blood cells have the highest radiosensitivity in humans. The bone marrow, spleen, and lymph nodes very sensitive to the action of ionizing radiation. Hematopoietic organs. Epithelium of gastrointestinal tract, also very sensitive. Study of the activity of physiological systems, in particular nervous one, showed the high sensitivity of central nervous system to the action of even small doses of radiation on the body. Bone and muscle tissues not least sensitive to the action of ionizing radiation. They are the most radioresistant. Cellular radiosensitivity is integral characteristic of cell that determines the probability of its death after exposure to radiation. Radiosensitivity is synonymous with radiosusceptibility. The term radiosensitivity is also used in relation to tissues, organs, and organisms as a whole, biological species, and etc. With equal radiosensitivity of biological objects, the degree of their damage is determined primarily by the radiation dose. Already in the period of early radiobiological observations, which were predominantly descriptive, it became obvious that damaging effect of ionizing radiation on biological objects is dose dependent. Construction of dose effect graphs made it possible to compare the radiosensitivity of biological objects, comparing radiation doses that cause equal effects on them, and to evaluate the ability of certain substances, radiomodifiers, to change the radiosensitivity of biological objects. The ratio of radiation doses that cause the same effect in biological objects in presence and in absence of radiomodifier is used as the main indicator of the radiomodifying effectiveness of substance. This indicator called the dose change factor. Mechanism of biological actions of ionizing radiation. When exposed to ionizing radiation of biosubstrate, the latter absorbs the radiation energy. This leads to the ionization of atoms and molecules. That is the loss of electron by atom. The remaining part of atom acquires a positive charge, or to the excitation of atoms, molecules, the transition of electron to higher energy level within an atom. The ions and free electrons formed as a result of ionizing of atoms interact with each other and with surrounding intact atoms and molecules, forming free radicals. Inorganic radicals are formed during the radiolysis of water. Hydroxyl radical, hydrogen radical, hydrated electron, hydrogen peroxide, hydroperoxide, superoxide anion radical, atomic and singlet oxygen. The products of water radiolysis have a strong oxidizing effect on the organic substances of biological tissues. Under the influence of ionizing radiation and water radiolysis products, radiolysis of the molecules of the main biologically important organic substances of the cell, DNA, nucleotides, amino acids, proteins, carbohydrates, etc., occurs with the formation of organic radicals that have a damaging effect on tissues. In the presence of oxygen, organic radicals interact with it. High affinity of oxygen for organic radicals underlies so-called oxygen effect, essence of which to enhance radiation damage to proteins, DNA, and other biomolecules in the presence of oxygen. The products of water radiolysis, reactive oxygen species and organic radicals, in turn are capable of changing the structure of biologically important macromolecules with violation of their function. The processes occurring in proteins, DNA, and phospholipids are of the greatest importance for the further fate of the irradiated cell. The structure of proteins is disturbed. As a result, secondary and tertiary structure of proteins changes, which leads to violation of their biological properties, including enzymatic activity. Radiation damage to DNA manifests itself in the form of violations of structure of nitrogenous bases, the appearance of DNA breaks, DNA protein crosslinks, and violations of DNA complexes with other molecules. In DNA damage, the role of the oxygen effect is great. In the presence of oxygen, the number of DNA base damage increases in more than 3 times. As a result, the genetic apparatus of cells is damaged, chromosomal aberrations occur, and processes of division and reproduction are disrupted. The description of phospholipids, of the lipid bilayer of cell membranes, the intensification of lipid peroxidation reactions contribute to the description of the structure and function of cell membranes and intracellular structure, their viscosity, permeability, and many physico-chemical characteristics. As a result, a number of functions of biomembranes vital for the cell disrupt. Barrier, receptor signaling, regulatory, transport, etc. Numerous structural and functional changes in subcellular structures cause disruption of oxidative phosphorylation processes, which leads to the depletion of energy resources of cells, release and activation of hydrolytic enzymes of lysosomes, inhibition of DNA synthesis and cell division, perversion of ion transport, and etc. Numerous damage to macromolecules and functional disorders significantly affect the viability of irradiated cells and tissues of the body, the cause of their damage and repair processes, and lead to the cell death. There are two forms of radiation cell death: interface and reproductive. Reproductive cell death is characteristic of all rapidly proliferating tissues. It occurs not immediately after radiation, but during several cycles of division. Structural DNA damage is the main cause of reproductive death. Interface cell death occurs before the cell enters mitosis. For most cells of somatic tissues of animals and humans, interface death is recorded at high doses of radiation, manifests itself in the form of various degenerative changes in the nucleus and cytoplasm, and occurs either directly under the beam or in the first hours after radiation. The exception is lymphocytes, for which the interphases type of post-radiation death is the main one and is registered already after irradiation at doses of 1 or 2 grays. The molecular mechanisms of interface cell death based on damage to cytoplasmic, nuclear and mitochondrial membranes, leading the disruption of water and electrolyte balance, release of hydrolytic enzymes for lysosomes, destructive changes in the nucleus, and etc. At high doses of radiation, a large number of cells die due to metabolic disorders. Morphological and functional changes occur in tissues, organs, and systems. So the events that occur in biological systems during and after exposure to ionizing radiation conditionally divided into 4 successive stages. The first, physical stage. Within a very short period of time, within 10 minus 13 seconds, Energy of ionizing radiation absorbed by biomolecules and components of their environment, molecules of water and other substances. As a result, excited and ionizing atoms and molecules appear. Second, physico-chemical stage. Due to the excitation and ionizing of molecules, internal rearrangements occur in them due to migration of energy and charge within the molecule or between molecules. There are initially damaged biomolecules and their degradation products, ions and chemical radicals. Approximately 50% of all emerging radicals formed from water molecules, which make up about 70% of the mass of living organisms. The third, chemical stage. Ions and reactive radicals at the place of their formation and some distance from it. 300 or 350 nanometers on average, attack by molecules, damaging them. It also happens for a short period of time. As a result, secondary chemically damaged biomolecules appear. And the fourth biological stage, the terms of formation, this stage a wide range from 10⁻⁶ seconds to many years. Processes developing during this period based on change in intracellular intermediate metabolism. Depending on the dose of ionizing radiation received, intracellular metabolism is disturbed to varying degrees, from small shifts with subsequent recovery to complete and persistent breakdown, leading to cell death. Completion to the stage of biological reactions in people who have undergone an acute stage of disease are long-term consequences, somatic and genetic, and early aging in the body. So, radiosensitivity of cells at different stages of cell cycle. The eukaryotic cell cycle consists of two periods. The period of cell growth, interphase, during which DNA and proteins synthesized and preparations made for cell divisions. It consists of several stages. G1 phase. G is mean on English gap, or the phase of initial growth during which mRNA, proteins, and other cellular components are synthesized. S phase, from English as synthesis, during which the DNA of the cell nucleus is replicated. Doubling of centrioles also occurs, if any. G2 phase, during which there are preparation for mitosis. Differentiated cells that no longer divided are in the G0 resting phase, having the same amount of DNA as in G1. The period of cell division, phase M, from the word mitosis, includes two stages: karyokinesis, nucleus division. Mitosis in turn divided into 5 stages: cytokinesis, divisions of the cytoplasm. Progression of the cell through the cycle is determined by the activation of successive protein kinases activated by the cell cycle proteins, cyclins. Protein kinases phosphorylate proteins and thereby change their conformation and enzymatic activity. There are several so-called checkpoints in the cycle. Checkpoint control post, during the passage of which the enzymatic systems check DNA for damage, and if they are detected, inhibitors of cyclin-dependent kinases are activated, which slows down the transition of cells from one phase to another. This slowing down of the transition gives more opportunities for DNA damage repair. When significant amount of damage is applied to the cell, for example, radiation damage, the cycle is delete, division block, mitotic block to ensure DNA repair. The most sensitive are in differentiated cells that are well supplied with blood, divided rapidly and have active metabolism. In the human body, such cells are gametes, erythroblasts, epidermal stem cells, and stem cells of gastrointestinal tract. Neurons and muscle cells have minimal sensitivity. Other sensitive cells include oocytes and lymphocytes. The reasons for their sensitivity are not clear. Tissue radiosensitivity is a relative concept. In radio-resistant stationary or weakly proliferating organs and tissues under the influence of irradiation, hidden typical radiation damage occurs. In particular, chromosomal aberrations, which can be detected under conditions of activation of cells divisions, for example, in the processes of post-traumatic regeneration. Radiation injuries that develop in the long term after irradiation due to the death of functional cells of weakly proliferating tissues, such as blood vessels, bones, and nerves, are late deterministic effects. Organ radiosensitivity depends on the radiosensitivity of the tissues that form this organ. Criteria the mass of the organ decreases, the functional activity decreases, muscle weakness in case of acute radiation sickness, devastations of the organ by specific cells, site of tumor when the lungs irradiated with 6 degrees. Classification of organs by radiosensitivity. The most radiosensitive, there are lymphoid organs, red bone marrow, gonads, and small intestine. The average degree of radiosensitivity: skin, endocrine glands, and radio-resistant organs. There are liver, kidneys, and brain. So let's see the radiobiological effects in the region of low doses. Is about 0.01 to 0.1 grays, and radiation in low doses causes DNA damage of all types: single strand breaks, double strand breaks, modifications, damage or loss of the base, DNA cross-links, and DNA protein. The vast majority are single-strand breaks, most of which are repaired by living cell within a few minutes after radiation exposure, and almost all the rest, with a few exceptions, within a few hours. Until the 1990s, the sensitivity of available methods was not enough to reliably detect DNA damage of various types after low-dose irradiation, even in relation to the simplest and most massive type, single-strand breaks. However, by the end of the 1990s, there was extraordinary increase in the sensitivity of the technique due to the development of a method for detecting double-strand breaks by the number of 4C of the histone, and somewhat later the 53BP1 protein, which can be detected by antibody fluorescence. This made it possible to determine the double-strand breaks after irradiation, as mentioned above, even at very low doses, up to 10 milligray. Not to mention of low dose range, where a linear dose dependence was demonstrated. Thus, the range of low doses is a zone of confident fixation of effects or in terms of the release of double-strand breaks, although some authors express doubts about an ambiguous relationship between detected fluorescent foci of repair proteins and unrepaired double-strand breaks. Double-strand breaks are one of the simplest types of most dangerous DNA damage, cluster or complex, which are characterized by hindered repair. Cluster effect occurs when ionization damage sites are distributed along the DNA molecule into compact clusters of complex, so-called complex damage. Singular effect is more inherent in densely ionizing radiation than in rare ionizing radiation. But for the latter, there are also significant differences in the effect of radiation compared to spontaneous DNA damage by free radicals. The number of clusters directly depends on the radiation dose. Complex DNA damages cluster differ in their ability to be repaired by the cell. Thus, initial level of radiation double-strand breaks weakly correlates with a subsequent biological effect. Remaining unrepaired double-strand breaks correlate with this effect, although the number of such double-strand breaks may be very small. Among the general pool of double-strand breaks, various classes are distinguished, differing in their ability to repair. As a result, hypothesis appeared according to which it is damage clusters, complex damage, that are responsible for lethal and or mutagenic, carcinogenic effects of ionizing radiation. Theoretically, a certain amount of DNA cluster damage should be induced by arbitrarily small doses of radiation. Indeed, the yield of complex damage in human cells exposed to X-ray radiation almost strictly linear depends on the dose in the ranges of both medium and high and low doses. In the latter case, when cells are irradiated under conditions that include DNA repair. Induction of cluster or complex damage, main factor determining the specificity of damaging effect of radiation. Even simplest complex damages induced by radiation no less than 10 in 5 more often as a fraction of the sum of all DNA damage that then occur in the processes of natural cellular metabolism. The potential molecular mechanism of low-dose mutagenic effect via double breaks induction has been demonstrated in a number of subjects in vitro and in vivo. The latter include experiments on mice, minimum doses of 10 and 100 milligrams, and on lymphocytes after computed tomography, in which cells irradiated at doses of the order of 20 to 100 milligrams. It was found that 5 minutes after the end of the diagnostic procedure, lymphocytes recorded recorded double breaks at level which compare to effect of irradiation of the cells in vitro at doses of 20 milligrams. It should be noted that DNA damage index after computer tomography rapidly decreased to almost the initial level due to DSB repair. And only after exposure to dose of 100 milligrams, increase was still significant, even the maximum time studied of this account, 30 minutes after X-ray irradiation, accompanying the tomographic examination. On the other hand, the recent years, it has been reported that double break repair is difficult when cells irradiated in vitro in low dose range. However, many authors do not confirm this. If in the first group of works X-ray radiation prevailed, then in the second, gamma radiation. In general, should be said that range of low doses is the zone of confident registration of double breaks. The question of their reparation in this case is ambiguous. In this regard, potential molecular mechanism of the carcinogenic effect, formally speaking, is present. Again, should be repeated that the latter is not necessarily realized at the epidemiological level. DNA repair, hyperradiosensitivity, and induced radioresistance. For the range of low doses, in general, there are more reliable data on the induction of DNA repair than for very low doses. This is the reason for the one of the mechanisms of the radioadaptive response and the phenomenon of hyperradiosensitivity, induced radioresistance, when on the dose effect curves, in terms of cell survival and other parameters, such as, for example, release of double breaks in recent year, after the initial decline, which is believed to due the insufficient background level of genome damage to induce DNA repair, is followed by rise caused by inducible operation of systems for repairing the damaged DNA structure, induced radioresistance. Radioadaptive response and hormesis. Radioadaptive response clearly stimulated by low doses of radiation. Likewise, numerous hormesis effects on experimental subjects in vitro and in vivo, as well as individual epidemiological data for humans, have been demonstrated especially clearly for the low dose region. Cell death during divisions, reproductive death. As in the case of the very low dose range, such death at low doses also possible, but again not typical. Death of some cells, including exposure to low doses and even at doses at the beginning of the medium dose range, determines mechanisms of hyperradiosensitivity phenomenon discussed above. In general, reproductive cell death for low doses in relation to normal cells is not relevant. Hereditary genetic effects. Transgenerational effects, disorders and pathologies in non-irradiated children of exposed parents in relation to humans have not been shown in more than 60 years of research at any doses. In experiments on laboratory animals with no defect in DNA repair and reduced radiosensitivity, these effects begin to appear only when irradiated in medium doses. So that's all about low doses. So I think this is the end. And if you have any question, you can write it on the chat, and I'm trying to answer on that. And also, if you have any question about this lecture, you can send it on my email that you can see on this slide. Thank you for your attention. Okay, colleagues, I see that you have no question. If you want to get question, I see. Okay, is there any food what we have to consume to reduce the biological effect? Maybe a question out of topic. Okay, any food to consume reduce the biological. Okay. I'm trying to answer to your question. Thank you for your question. I think we have no some special food or product that reduce biological effect of radiation. If you see some scientific articles about this, we can see that there is no effects of products after irradiation, for example, or if I know only one medical component that people used, but only in the such case like such case like very big accident on the NPP, for example. This is iodine tablets, but in the normal life, the iodine tablets not will be helped to reduce effect. And our cells of our body, cells of our and not our cells of animals body and plants, organism them itself will be protecting from irradiating, because our DNA, our DNA can repair themselves if we have single break of DNA, and it is a temporary effect, and no one food or product not will not have the influence on the radiation of the irradiation effects on the organism. Thank you so much. Oh, you're welcome. If you want to get this presentation, you can write Mr. Denis Agafonov, and he will send you this presentation. And if you have any question about radiobiological effects and radiobiological articles about these effects, low doses, high doses, experimental data, If you want to get it, you can write me or Mr. Denise, and we will send you if you're interested in this theme, this topic. Thank you for your attention, and goodbye for everyone. Thank you very much.